Junichi Sadoshima from the Rutgers, New Jersey, United States, and also Dr. Adam from University of Alabama at Birmingham, USA. So before we start the webinar, so we would like to invite Professor Badrul Hagar as the director of IMERI to have an opening session from this webinar. Please, Professor Badrul Hagar.
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good evening and also morning as well. Welcome to all participants to the webinar in commemoration of World Heart Day. The Honorable Speaker, Professor Junichi Sadoshima, Medical Doctor Ph.D. from the Rutgers, New Jersey Medical School, United States, and also Associate Professor Adam R. Wende, Ph.D. from University of Alabama, Birmingham, USA, and also our head of the Metabolic, Vascular, and Aging Research Center IMERI FMUI, Dr. Diki Tahapari, and our panelist and moderator, Dr. Bambang Widyantoro, Ph.D., and also Dr. Agian Jeffilano, Ph.D. Okay, from they're all two also from the research, the Cardiovascular Research Group, Metabolic, Vascular, and Aging Research Center IMERI, Faculty Medicine, Universitas of Indonesia. Currently, many people ask about what is the mechanism of diabetic cardiomyopathy, and also there's question about what is the potential therapeutic strategy in diabetic cardiomyopathy. Several publications show us that the pathogenesis and clinical features of diabetic cardiomyopathy have been well studied in the last decade, but the effective approach to prevent and treat this disease are still limited. The evidence from preclinical and clinical study that might provide guidance for the development of targeted strategies. They also highlight some of the novel pharmacological therapeutic strategy for the treatment and prevention of the diabetic cardiomyopathy. An in-depth understanding for the pathogenesis of diabetic cardiomyopathy might provide guidance for the development of strategies to prevent and treat cardiovascular disease in patients with diabetes. So I hope we will take advantage of this webinar. And of course, we are very proud to have both prominent speakers of today's webinar. And as IMERI, we hope we can start to build a comprehensive collaboration in this issue in the next few years. So please enjoy the webinar. Thank you very much. Terima kasih, Pak Agian.
Okay, thank you very much, Professor Badrul Hagar. Okay, so we would like to start the webinar. So I would like to introduce our panelists first. Dr. Bambang Widyantoro. He got after getting the MD and PhD in Universitas Indonesia and also the Kobe University, Japan. He is currently the principal investigator of cardiovascular division in Metabolic, Cardiovascular, and Aging Cluster IMERI. He is also the lecturer in Department of Cardiology, Cardiovascular Medicine in our university. And then after that, I would like to introduce our first prominent speaker, Professor Junichi Sadoshima. So after got the MD and also PhD in cardiology in Kyushu, he went to the Beth Israel Hospital in Boston to get the postdoctoral fellowship. And currently, he is the professor and also the chairman in the Department of Cell Biology and Molecular Medicine in Rutgers, New Jersey Medical School in the States. So his main research is mainly about the elucidate the underlying mechanism of heart failure and also the aging, including the autophagy, mitophagy, and also the mitochondrial dysfunction. So today he will give a talk about the autophagy or mitophagy and also the mitochondrial function in diabetic cardiomyopathy and also the cardiac aging. Thank you very much, Professor Sadoshima. So please start your presentation. Thank you.
Thank you. do you see my slide?
Yes, please.
Okay, great. So thank you very much, Jeffy. And also, it's a great honor for me to present my work in this conference. And my lab is focusing on various mechanisms promoting cardiomyopathy, including diabetic and obesity cardiomyopathy. And I would like to focus my talk on regarding autophagy and mitophagy. And since this is a relatively specific subject, so I try to be more general and try to explain the role of autophagy and mitophagy in heart failure in general, and include their role in cardiomyopathy in the middle. So autophagy is an important mechanism of cellular degradation of protein and organelles, characterized by the presence of this double membrane structure, autophagosome. And the content is degraded by lysosome. And this is a really critical mechanism for the cellular quality control mechanism. And also the content can be recycled after it becomes amino acid and fatty acid, so that it can also produce ATP. And today, I would like to talk about the role of autophagy in heart failure in general. And also, autophagy is generally very non-specific mechanism, but it can target protein and organelle. And when autophagy target mitochondria, we call it mitophagy. And we recently found that mitophagy is very important for the preservation of heart function in obesity and diabetic condition. And also, this is a little bit, different from the main subject of this symposium, but I would like to tell you that autophagy is also can be dysregulated. So we should understand autophagy can be toxic in some condition. I mentioned it. And finally, I would like to discuss some therapeutic implication of autophagy. So the reason why we became interested in autophagy almost 20 years ago is we have a myocardial hibernation model, and hibernation is amazing mechanism for the cardiomyocyte myocardium to survive even in the presence of severe ischemia. And we were so curious what happens, why myocyte can survive even in the presence of severe myocardial ischemia. And we look at proteomics and genomics. And so basically we conducted unbiased screening. And the most significant mechanism upregulated in hibernating myocardium was autophagy-related mechanism. So we looked at the myocardium by EM, and we found that the many autophagosomes were observed in ischemic myocardium. And we are very excited because at that time, we don't know much about autophagy, and look at the literature, and it described that it is a mechanism to preserve ATP and also control protein and organelle quality. So we decided to pursue this mechanism, and we switched the model from pig to mice because we can do many genetically manipulation. So we first created a permanent coronary ligation mouse model so that we can induce ischemia and myocardial infarction. And using autophagy indicator mice, we found that autophagy is induced in border zone. We see many autophagosome in border zone. And we found that if you use heterozygous pectin knockout mice, which can reduce the level of autophagy like this, we found that the accumulation of dysfunctional protein increases, and also they have higher mortality in chronic phase, and also the infarct size is actually chronically increased. So we concluded autophagy is very important to eliminate dysfunctional protein and improve survival in chronic phase, and also involved in infarct resolution in chronic phase, so that if you don't have autophagy, infarction size is increased like this. So, but the patient have to live, even after chronic condition. So, that's why autophagy is activated, but probably autophagy is not enough, and the patient eventually develop heart failure. So we speculate that there might be some negative regulators. We believe that the autophagy is not enough in patients. So we looked for what kind of mechanism regulate myocardial autophagy, and so far we found that the one protein kinase called MST1 is very important, suppressing autophagy. This is actually originally found as a kinase to induce cell death, but we found that this MST1 activation by stress markedly strongly suppress autophagy, and that contribute to the development of cardiac dysfunction in patient. And in terms of mechanism, the autophagy is regulated by various molecule termed ATG, autophagy related genes, and BECRIN1 is one of them. And it has been shown that this BECRIN1 is inhibited by BCL2 binding, as you see here. But interestingly, this MST1 promote interaction between BCL2 and BECRIN1, and autophagy is suppressed. Actually, MST1 phosphorylate one residue in BECRIN1, and that actually promote protein-protein interaction, as you see here. So to test the relevance of this observation, I have a former fellow in Taiwan. He's a cardiac surgeon, so that he has access to a failing heart. And we found that MST1 is activated, many cases in heart failure heart, and BECRIN1 is phosphorylated, and in those heart, the autophagy is strongly suppressed. So we believe that MST1 activation is very important, and inhibitor can be considered for heart failure treatment. And if we have a loss of function MST mouse model, and in fact that those mice have improved survival after myocardial infarction. But if we cross this loss of MST1 function mouse model with autophagy deficient mouse model, we no longer observe advantage in survival. So we believe that this survival advantage of MST1 loss of function model is mediated by stimulation of autophagy. And as I mentioned, autophagy usually degrade cellular content in a non-specific manner, but it has also a unique mechanism to degrade damaged portion of mitochondria. This is really a cool mechanism because when a part of the mitochondria is damaged, that portion is selectively recognized by autophagy machinery, and that portion is separated and can be degraded. And it turned out that a cell has several mechanisms mediating this mitophagy. And the most important one is you may have heard of PINK1 and parking dependent one. And when PINK1 is activated in mitochondria, parking, it's a E3 ubiquitin ligase, actually ubiquitinate outer membrane proteins in damaged mitochondria. And then that E3 ubiquitinated protein can be recognized by autophagosome. But besides this mechanism, some receptor also exists, and somehow this receptor is expressed in a damaged mitochondria, and mitochondria is degraded. But you should keep in mind that the mitochondria in the heart is very tightly packed in between myofibers. So these mechanisms primarily discovered in fibroblasts and other cell type may not be necessarily important in cardiomyocyte or mitophagy. So that's why we are actively investigating mitophagy mechanism in the heart. So in terms of clinical implication, we have first used pressure overload model, and in mice, by ligating transverse aorta, we can impose pressure overload in mouse heart. And we look at mitophagy activation, and it actually, pressure overload relatively quickly activate mitophagy. But unfortunately, this mitophagy is very transient. This is a general feature in myocardial disease. I will show you the example in diabetic cardiomyopathy later, but mitophagy can be activated, but it's always transient. And after that, mitochondria dysfunction become obvious, and then development of heart failure follows. So it's a very important mechanism, compensatory mechanism, but unfortunately it does not last long. So we have a way to reactivate mitophagy, and one of the best intervention is TAP-BECLIN-1. This is a short peptide, and it can mobilize the autophagy inducer BECLIN-1 from intracellular storage site. And we are able to show that this is a mitophagy indicator mice, and in the presence of TAP-BECLIN-1, we can activate mitophagy even in the fading heart. So we treated mice with a TAP-BECLIN-1 and gave a pressure overload, and we are able to rescue cardiac dysfunction in a model. But this rescue is not cannot be observed when we inhibit autophagy or mitophagy in various genetically altered mouse model. So we believe that reactivation of mitophagy is a promising way to delay development of heart failure in patients. So then what happened to diabetic cardiomyopathy? And our group utilized high-fat diet. It's obesity and type 2 diabetes mouse model, and 60% saturated fatty acid will be fat. And after 3 months, we see increases in body weight and in insulin resistance and diastolic dysfunction. This is you may know the HFpEF also is characterized by this mechanism. And cardiac hypertrophy, not severe, but the enlargement of my cardiomyocyte is also observed. And also inflammation, Dale Abels and others propose that inflammation is a very important mechanism. So these are induced. So what happened to autophagy? And we found that the autophagy is actually activated. And you may have heard of this LC32 is indicator. And very interestingly, we found that the LC3 is mostly observed in mitochondria. So if you give high fat, cell mobilize the mechanism of autophagy, focusing on clearance of mitochondria. So this alone probably indicates that the mitophagy, maintaining mitochondria function is very, very important. And we have a way to visualize autophagosome, green fluorescent protein. And we also found that this ring structure and puncta always co-localized with mitotracker. Sorry, this that does not show well in my slide, but the LC3, most of LC3 co-localized with the mitochondria. But as I mentioned in pressure overload mouse model, this mechanism doesn't last long. So if you look at LC3, it reached the peak 6 to 6 weeks, around 6 weeks. But by 20 weeks, the level comes back to the normal level. And more importantly, we have to measure the autophagic flux by comparing control and lysosome inhibition condition. If you do not see further accumulation of LC3, we must say that the autophagic flux is stopped. And we found that by 20 weeks, autophagy is no longer activated. But if I use marker of mitophagy, surprisingly, we found that the mitophagy, in case of obesity cardiomyopathy, mitophagy is activated even after conventional autophagy is inhibited. And we can easily observe autophagosome containing mitochondria by electron microscopy, as you see here. So we are very curious why this happens. And we subsequently discovered that mitophagy is mediated not only ordinary autophagy mechanism, but also unconventional alternative mechanism, which does not require conventional autophagy machinery. And we call it alternative mitophagy, and we believe that the Lab9 and ULK is important. So then we have a way to inhibit this alternative mitophagy, and we found that this actually, the activation of this alternative mitophagy is really, really important for the chronic condition. And people are focusing on ordinary mitophagy regulated by conventional autophagy machinery and PINK1 parking, but this mechanism is not activated in chronic phase, but the alternative mechanism is activated. And if I inhibit this alternative mechanism, we saw enhancement of cardiomyopathy, and we saw mitochondria structure is disrupted, you see here, and lipid accumulates. Most likely fatty acid oxidation is inhibited. And we have a way to stimulate this alternative mitophagy. We are able to slow down the development of cardiomyopathy. So I can say mitophagy is not enough, but certainly the maintaining this compensatory mechanism is very important. So basic field, most researchers are focusing on mitophagy by conventional mechanism, but in terms of clinical relevance, you know, we really have to find out what's the mechanism of mitophagy activated during chronic phase of disease, which does not rely on conventional mechanism. So we are very actively studying the mechanism of mitophagy activated by even in the absence of conventional mechanism. So I switched my talk to autophagy dysregulation. I think this is important because, you know, judging from what you may think that autophagy should be always stimulated, but the clinical case that is not the case. So I just want to tell you one example. And we recently discovered that autophagy dysregulation can cause cell death in cardiomyocyte. And this is based upon Dr. Beth Levine's group observation 2013. When autophagy is dysregulated, it can cause cell death. And very characteristic morphological feature include presence of many vacuole and also condensed mitochondria, and also very characteristic nuclear morphology. There's a space next to the nucleus. We call it the perinuclear space. And also, Dr. Levine's group did the screening, compound screening, and the cardiac glycoside can very specifically inhibit this autophagy cell death. And they named it autosis, and autosis is characterized by both morphological and biochemical features. And we look at whether this is observed in a heart, and we found that this autophagic cell death is observed during ischemia reperfusion, late phase of ischemia reperfusion. We see very characteristic perinuclear space, as you see here. But the time course is very slow compared to ordinary cell death. As you may have heard of, the ischemia reperfusion usually kills myocyte within first few hours, within 1 to 2 hours. But this autotic cell death occurs during the late phase of ischemic reperfusion. and also sensitivity to cardiac glycoside is one feature. And we found that the warfarin can inhibit autosis and also ischemic perfusion. But one thing I have to mention is that warfarin can inhibit sodium potassium ATPase, and but this is not very effective in mice, mouse heart especially. And so we have to use humanized mice in order to do this experiment. Since human sodium potassium ATPase is more sensitive than mouse sodium potassium ATPase to warfarin treatment. So we got knock-in mice from Dr. Levine, and then we conducted this experiment. And in fact, this warfarin treatment can inhibit autosis and inhibit myocardial injury. And we examined the mechanism of the autosis, and we found that the two molecules very significantly upregulated during the late phase of ischemic perfusion. One is a Rubicon, another is a TFEB. And we initially thought that autophagy is strongly activated, and that actually degrade many things and kill the cell, but it was not the case. And because the Rubicon inhibit autophagosome lysosome fusion. And so autophagy never goes through at the late phase of ischemic perfusion. But with activation of a TFEB, we found that this actually stopped lysosomal fusion, induces accumulation of autophagosome, and the activation of TFEB further stimulate autophagosome, so that that's why autophagosome accumulate in at the pathological level. And more importantly, when autophagosome is formed, autophagosome membranes are made by intracellular membranes. So in order to make autophagosome, you have to use mitochondria and ER membranes, and those membranes are usually recycled. But this recycle process is attenuated, and so in this condition, making further autophagosome is very, very toxic. And in fact, that we saw disappearance of mitochondria and ER in this condition. So we created the Rubicon knockout mice so that we basically improved recycle of autophagosome membrane, and we are able to show that it can inhibit autotic cell death, and also we can rescue the myocardial injury, as you see here. So we believe that there are important, important recognition regarding autophagy dysregulation. In a developmental biology, some condition, autophagy actually degrade many things and kill the cell. But in pathological condition, autophagy block in the between autophagosome and lysosome, that actually causes a marked accumulation of autophagosome, and then that causes cell death. So to summarize my talk, sorry I presented many things, but I presented autophagy is a very important mechanism in the heart, and in some pathological condition, autophagy activation is not enough, and that stimulate heart failure. And mitophagy is the same thing. You know, the pressure overload and diabetic cardiomyopathy, maintaining mitochondrial function is very important. And it is activated, but the conventional mechanism is only transient, and the chronic mechanism, alternative mechanism exists, but certainly it's not enough to prevent development of heart failure. So in clinical condition, we have to identify the level of autophagy and mitophagy, and we have to intervene. We have to supplement autophagy so that we can prevent development of cardiac dysfunction. But in some condition, autophagy is dysregulated. And although autophagic flux may not be necessarily activated, but TFEB can stimulate marked increase in autophagosome. And in that condition, we have to have an intervention to normalize the level of autophagy. So main focus of the symposium is diabetic cardiomyopathy, but I define cardiomyopathy, diabetic cardiomyopathy, obesity cardiomyopathy is a condition where the mitophagy is not enough. So we really have to find an intervention to normalize this condition. So finally, I want to introduce my fellows, and also I have a very good collaboration with Dr. Bess Levine, who passed away 2 years ago through the Duke Foundation Network. Thank you very much for your attention.
Thank you very much, Professor Sadoshima. So very wonderful and also insightful lecture about the role of autophagy and also mitophagy as the alternative one. So thank you very much. So now, so we would like to invite our second prominent speakers, Dr. Adam. So Dr. Adam is the associate professor in the Department of Pathology from the University of Alabama in Birmingham. So after got the Ph.D. in the Washington University, he continued his postdoctoral fellow in University of Utah, and also he is currently as the associate professor in the University of Alabama. So his main research is about the molecular pathway mediating metabolic switching, including in diabetes. And today, we would like to give a talk about the switch challenge, how glucose reprograms cardiac metabolism in diabetes. Thank you very much for joining us, Dr. Adam, so you can share your presentation.
Okay. There we go. Okay, can you see it just fine?
Yes, perfect.
Perfect. Well, I'd like to thank the organizers for this invitation and for including me in this great day to honor World Heart Day and talk a little bit about some of the mechanisms in diabetic cardiomyopathy. And I really enjoyed the first talk, and I hope I can follow and add some additional perspectives into other mechanisms that may be at play in diabetic cardiomyopathy. And I have no disclosures. So over the next 20 minutes or so, I'd like to break my talk down into 3 main areas. One, to really discuss how cardiac metabolism and its flexibility or inflexibility in the disease state may impact the development of heart failure. And then introduce two additional mechanisms that we haven't heard about today. One is a post-translational modification called oglycinylation, which I'll get into quite a bit detail later. And the other is transcriptional reprogramming through epigenetics, both of which I think have a component that's being mediated by glucose, not as a mitochondrial substrate, but as a signaling molecule. And then I'll finish with some unpublished human data, kind of suggesting some of the relevance of this. So I know this audience doesn't need that long of an introduction, but I do want to put a few things in perspective. We do know that in addition to the pandemic of COVID, there's another pandemic, and that's of diabetes. And really with many countries showing and regions of the world showing double and even triple digit increases in the next 20 years or so. And although I'd love to be here in Indonesia with you right now giving this talk in person, I'm actually way over here in Alabama. And one of the reasons for my interest in this is Alabama, of the states in the US, is one of the hardest hit by obesity, diabetes, and heart disease. And unfortunately, we're often ranked in the top 3 and often even in the top 2 for these horrible diseases, for which we know there's quite a bit of correlation. But one thing my lab and several other labs, as we've just heard, is trying to find out what is the relationship to nutrient excess that's seen in obesity and diabetes and its relationship to the development of heart disease. And there are lots of mechanisms looking at this. It's been looked at for several decades, well over half a century, and maybe even longer. So there's a lot of potential mechanisms. And as we just heard, one of those potential mechanisms is disrupted autophagy. But where my lab chooses to focus is this idea that the impairment of energy substrate utilization in the heart is driving several of the mechanisms leading to heart failure associated with diabetes. Now, many investigators have suggested that the heart is this metabolic omnivore, utilizing whatever fuel it's given, because it is this engine that needs to be constantly generating ATP to keep up contraction and blood flow throughout the body. And the healthy adult heart relies primarily on fatty acid utilization, but as substrate selection and availability changes, this utilization of the heart to keep up ATP is also changing. But what I want to propose is that these different substrates are not only important to the heart for generating ATP, but that disruption in these different levels actually drives cellular signaling that further impacts the development of disease when in the chronic state. Now, this idea of metabolic, inflexibility is not a new idea. It's been studied for quite some time. And going back well over half a century, it had already been shown that in the context of diabetes, in both humans and canines, there was this mismatch of substrate availability and utilization, where even in the context of hyperglycemia in diabetes, there was a relative reduction in glucose utilization in the heart. Now, what I did not realize until starting my faculty position here in Birmingham was that this work was actually done right here at UAB all those years ago. So it's kind of nice to come full circle to where some of this work started. But what does this actually mean for the heart and the development of diabetic cardiomyopathy? Well, what it means is when the heart is unable to readily switch between these different fuels, several different pathways get backed up, especially when kept in the chronic state or this metabolic rigidity in the diabetic heart. And what this ends up doing is actually further enhancing fatty acid utilization, but in an uncoupled and dysregulated way, leading to generation of things like lipotoxic metabolites like ceramides, or just myocardial steatosis or fat accumulation in the heart. But what this signaling also does is it leads to this disconnect between the fuel utilization and metabolic efficiency and starts shunting these nutrients into other pathways, such as the hexosamine biosynthesis pathway, or HBP, which can lead to protein post-translational modifications. So the overarching question in our group is, how do these changes in cardiac metabolic substrate flexibility affect the heart, and are they adaptive or maladaptive? And if they are maladaptive, how are they regulated so that we can reverse these? So we've taken a couple different approaches to look at these different substrate utilization pathways and to try and define the mechanisms leading to them. And one of the first questions was simply, is it the substrate delivery that's impacting disease progression in diabetic cardiomyopathy? And one of the reasons why we thought it was simply delivery of glucose to the heart was early work done a little over 20 years ago by Belke and colleagues, where they looked at a type 2 diabetes model of mouse, or the DB-DB mouse. And in this model, they showed that cardiac substrate utilization at the level of glycolysis or glucose oxidation was reduced in the heart. Furthermore, what they showed when they overexpressed the glucose transporter GLUT4 systemically was that they were able to prevent the development of this metabolic substrate decrease. So this led us to hypothesize that maybe simply restoring myocardial glucose delivery could attenuate development of this diabetic cardiac dysfunction. And to test this hypothesis, we generated an inducible and cardiomyocyte-specific GLUT4 overexpressor. So unlike the earlier work that was overexpressing GLUT4 from birth, we wanted to see if we could induce it in the heart after diabetes, and if we could cure the diabetic cardiomyopathy, and that if expression in cardiomyocytes was sufficient to do this. So this is the doxycycline or TET-on model, where we have alpha-myosin heavy chain for cardiomyocyte specificity. And when the ligand doxycycline is added to the chow, we can have overexpression of the second transgene, which is a MYC-tagged glucose transporter GLUT4. So shown here is a Western blot simply showing that in our double transgenic mice, there is overexpression of GLUT4 only in the presence of doxycycline. There's no leakiness. And this reaches about a 5-fold induction at about by 2 days, and we maintain it out to 2 weeks. If we isolate cardiomyocytes from these transgenic mice and look at the GLUT4 surface levels, there's very little in the absence of insulin, but consistent with known insulin-mediated signaling to glucose uptake by GLUT4, we can see that this is enhanced with insulin. Consistent with more GLUT4 at the surface of the cell, we see that there's increased glucose uptake into these isolated cardiomyocytes by 2-deoxyglucose uptake, similar to wild-type mice in response to insulin, which is further enhanced with the transgene. So we kind of have this dose-dependent response. And this is just quality control showing that our model works. So what about the ability of substrate delivery to rescue the heart? So to do this, we move to a different model of diabetes or hyperglycemia, the streptozotocin-induced hypoinsulinemic model. So in this case, we took 4 groups of mice, controls or those double transgenics, and injected them with either a vehicle or streptozotocin. By 2 weeks, all the STZ injected mice were diabetic, and at that point, we introduced doxycycline to enhance glucose uptake into the hearts by transgene overexpression, and followed the hearts out the mice for an additional 2 weeks. And at this 4-week time point is where we did all of our studies. So similar to the earlier analysis, we performed isolated working heart and looked at glycolysis and glucose oxidation. And consistent with our isolated cardiomyocyte studies, overexpressing GLUT4 was sufficient to enhance glycolysis and glucose oxidation. And similar to the Belke study, we were able to protect the dysfunction in glycolysis. However, to our surprise, we actually found that not only did we not rescue glucose oxidation in these mice, we actually further exacerbated the defect in mitochondrial oxidative phosphorylation. So this led us to revisit our hypothesis. And it turned out that enhancing or restoring glucose delivery to the diabetic heart not only did not attenuate development of diabetic dysfunction, it actually exacerbated or enhanced it. So now we've been trying to figure out what are the mechanisms of this cardiac glucotoxicity. And one of the first places we looked was that of mitochondrial function. So we went through and measured the complex activity of each of the OXPHOS complexes and found that enhanced glucose delivery alone was not sufficient to lead to mitochondrial dysfunction, as shown by the open circles in the MYC or MG4H. But in each context, when the mice were diabetic, or had STZ, and then we enhanced glucose delivery, we could see a decline in activity of each of these complexes.
Dr. Adam.
Oh, sorry, I muted when I cleared my throat. So what was the mechanism for this? One mechanism that we think is responsible is that of protein post-translational modification via O-glycanylation. So we all know glucose can be taken into cell and used for glycolysis, or can go down several other pathways, including the hexosamine biosynthesis pathway. And when it goes down the HBP, it generates this metabolite UDP-GLICNAC, which can be used to modify proteins, much like phosphorylation. However, unlike phosphorylation that has hundreds, if not thousands, of kinases and phosphatases, O-glycanylation is mediated by only two proteins, O-glycanac transferase, or OGT, to add it, or O-glycanase, or OGA, to remove it. And this post-translational modification has been studied in pretty much every organ and disease state and health state, and is critical to cellular function, really regulating protein function in much the same way that phosphorylation does, with the key difference being that it's mainly nutrient driven. So we looked back in our mouse hearts specifically at the mitochondrial proteome to see what was changing for the GLICNAC modifications, and we could see that the transgene alone could enhance mitochondrial glycanylation, STZ or diabetes could enhance glycanylation, and the combination further disrupted this. We further went through to identify which proteins were being modified and found that several mitochondrial oxphos enzymes were being O-glycanylated, and we and others have shown that this leads to a decrease in protein function when sustained in the mitochondria. However, I was also curious what other changes this pathway may be driving, and as a classically trained transcriptional biologist, I was wondering what was going on with the transcriptome. So we performed RNA-seq analysis in our transgenic mice, in our diabetic mice, and in the GLUT4 diabetic mice to try and determine what changes in gene expression associated with diabetic cardiomyopathy may be being driven specifically by the glucose component of diabetes. So what's shown here in this Venn diagram is that there are several genes in the upper circle that are regulated specifically by GLUT4 alone or glucose alone. In the bottom right, there are several genes that are being changed by diabetes, which would have both hyperglycemia and hyperlipidemia. And then this can be further disrupted when we enhance glucose delivery to the diabetic heart. But we were curious in the common pathways. And when we looked at those genes that were co-regulated by glucose or diabetes, they were mostly involved in fatty acid utilization and this other metabolite, ketone body metabolism. So when we look at these individual genes, we saw that there were quite a few OXPHOS genes that were decreased and quite a few other metabolic genes that would be negative regulators of glucose utilization, such as the pyruvate dehydrogenase kinase 4 were upregulated. So overall, this led us to conclude, and there was a nice commentary along with this paper, that the diabetic heart is actually decreasing glucose utilization to protect itself from nutrient excess. And that inadvertently, when we overexpressed GLUT4 in an attempt to restore metabolic control, we actually led the heart to this forced glucose toxicity, because excess nutrient, when sustained, can lead to modifications of things like mitochondrial proteins and disruption of transcriptional expression. So in this first part, I hope I've convinced you that although there is this associated decrease with glucose utilization in the diabetic heart, that restoring that is actually not the therapeutic way to go. So what is and what are the mechanisms downstream of this glucose toxicity? So a former postdoc in the lab, Manoja Brahma, wanted to interrogate one of these pathways, and that is that of ketone body metabolism. And what he did was focused on this one enzyme, BDH1, which is a rate limiting enzyme in ketone body metabolism. And actually, interestingly enough, if we go back to that paper from the 50s, this was actually already seen, or this disruption of ketone utilization was known back that long ago, but most of the literature is focused on this balance between fatty acid and glucose utilization. But I think it's increasingly becoming known that ketone bodies are also an important metabolite that the heart can use, particularly in response to things like starvation, uncontrolled type 1 diabetes, and one of the more trendy diets right now are the ketogenic diet. So what are ketone bodies? There are several of them, of which beta-hydroxybutyrate one of the most abundant, and it's often used. It's generated by the liver and is used in extrahepatic tissues such as the brain, and as we'll see in the heart. Now, this has become increasingly well studied, particularly because of some of the SGLT2 inhibitor studies and alternative metabolism that may be going on in the diabetic heart, as well as what's going on in non-diabetic heart failure. So about 6 years ago, back-to-back papers came out showing that in the failing heart, that the non-diabetic failing heart, that ketone body utilization is actually increased. But what we're showing is that in the diabetic failing heart, ketone body utilization is actually being decreased. So what is the role of this metabolism, and why is it different in diabetic cardiomyopathy than ischemic cardiomyopathy? So one hypothesis is that it has to do with some of this downstream glucose signaling from hyperglycemia that isn't present in the non-diabetic heart. So this enhanced glycoalkylation. So what Brahma did was compared both the GLUT4 data and the type 1 diabetes data that I've already shown you with some of our human data in type 2 diabetic failing hearts. So we have access to human heart collected during left ventricular assist device sampling, and we've examined that, and I'll get into that in a little more detail. But for this initial analysis, Brahma was really just interested in what are common pathways driven by glucose and that might be common between different types of diabetes in both type 1 mice and type 2 humans. And one of the top pathways that Brahma found consistent with our mouse work was that even in humans, there was this suppression of ketone body metabolism. And it wasn't only this BDH or rate limiting enzyme, it was other enzymes in the pathway, such as OXCT1, which is encodes SCOT, and this solute carrier, which is one of the uptake mechanisms for ketones into the cell. So we returned to our isolated working heart to see what's going on with ketone metabolism, and consistent with our earlier studies, again, Brahma saw that GLUT4 or enhanced glucose delivery could enhance glycolysis, but consistent with the changes in gene expression in the diabetic cardiomyopathy, we see a suppression of ketone body metabolism. So is glycoalkylation related? So to get back to this question and to remind you, GLICNAC is modified by only two enzymes, this OGT to add or this OGA to remove. Many groups have tried to overexpress OGT, but this leads to a cardiomyopathy in itself. So we turn to a slightly different model. There's a naturally occurring splice variant of OGA that leads to spontaneous diabetes in rats, and it leads to a dominant negative form of OGA. So using the same TET-ON system, we wanted to see, is GLICNAC signaling downstream of glucose uptake, independent of all the other things that glucose delivery may do, sufficient to drive a diabetic cardiomyopathy-like phenotype? So this is a Western blot showing that we can get overexpression of DNOGA and that this leads to enhanced glycoalkylation. We then looked at the expression of BDH and saw that it was decreased in these hearts, and that we further went on to associate that with the decrease in ketone body metabolism. So this is already published, so I'll just quickly summarize. In both type 1 diabetes models, glucose delivery, and enhanced glycoalkylation, all models that show disruption of this signaling, we see a decrease in ketone utilization, and this is consistent with our human failure model. So I know I'm going a little bit long, but I'm going to add one more story. And what we further went on to show is this sufficient to drive a diabetic cardiomyopathy-like phenotype? So my current postdoc, Dr. Ha, took that same DNOGA model and enhanced it to see, does sustaining glycanalylation, like would be seen in the chronic diabetic state, is that sufficient to lead to diabetic cardiomyopathy? So instead of looking at only 2 weeks of enhanced glycanalylation, she went out to nearly 6 months and showed that this could lead to an enhanced protein glycanalylation that really hit a maximum at about 2 weeks and didn't really increase that much more out to 6 months. But interestingly enough, it led to a progressive diabetic cardiomyopathy-like phenotype with development of cardiac hypertrophy, and not shown here fibrosis and decreased gene expression associated with mitochondrial diabetic cardiomyopathy. So I hope I've convinced you that in the chronic disease state, there's several different mechanisms that are going on. And we think that part of the dysfunction seen in diabetic cardiomyopathy, particularly when diabetes is left unregulated, is enhanced signaling through glycanalylation that alone is sufficient to lead to a diabetic cardiomyopathy-like phenotype. Now, one important point is that it alone is a very progressive and slow disease. And I think that just has to do with, this is only part of the story. We all know that diabetes is not just hyperglycemia, it's also hyperlipidemia. And I think that's part of the reason for this slow progression. Now, I think I'm out of time. Is that correct?
Yeah, but it will be fine. So,
okay. Yeah. Okay, so one other question that we really had was, is this leading to some of the increased susceptibility shown in the diabetic population? And we believe that part of this enhanced glycanalylation is driving epigenetic changes in the diabetic heart to enhance the susceptibility to disease. So epigenetics is just everything above the genome, and there's several different mechanisms. Modification of histones, modification of non-coding RNAs, and modification of DNA. And when DNA is modified, specifically through DNA methylation, it is inversely related to gene expression. And we and others propose that some of this nutrient signaling can actually lead to these epigenetic modifications that lead to that metabolic rigidity by disrupting gene expression associated with metabolic control. And we've shown, and this was published last year, that in cell culture at least, enhancing glucose delivery to cells can reprogram the DNA methylome consistent with changes in gene expression that show endothelial-like dysfunction consistent with diabetic cardiomyopathy through increased NOS signaling and decreased VEGF signaling. So what we've done is then went into that same mouse model to say, is GLICNAC driving this? So one thing I didn't tell you is that the GLICNAC machinery, OGT, interacts with the epigenetic machinery to reprogram the DNA methylome. So we wanted to see, was cycling GLICNAC, which would be consistent with poor glycemic control moving to good glycemic control, was that sufficient to alter susceptibility to heart disease? So we enhanced glycanalylation in the mice for 2 weeks, and then we restored GLICNAC levels to normal. And then, as we heard in the previous talk, we then subjected these mice, even when glucose levels and GLICNAC levels were back to normal, to transverse aortic constriction or pressure overload, and found that even though GLICNAC levels were restored to normal, there was some type of sustained susceptibility to pressure overload induced hypertrophy. And we're still trying to figure out the mechanism for this. And what we really wanted to see is any of this relevant to the human condition? So we went back to our LVAD samples and said, of the changes in gene expression in these diabetic human hearts, so shown here is the methylation pattern in non-diabetic human hearts and diabetic human hearts, which were associated with gene expression changes. And we used some pretty stringent bioinformatic filters to find only those genes that were differentially expressed inversely with DNA methylation. And shown here is those top 17 candidates. And consistent with our mouse data, BDH1 was one of the few genes that was hypermethylated and suppressed. But what we really want to know is, what's the mechanism? So as you'll note, shown here in the black is DNA methylation decreases, with gene expression increases. And one of the top demethylated and induced genes is GAD45, which also interacts with the O-GLICNAC machinery and is a gene responsible for DNA demethylation. So now we're trying to figure out, is this a common mechanism for metabolic reprogramming of gene expression? So I'm sorry I ran a little bit over, but I hope I convinced you that restoring glucose delivery to the diabetic heart is probably not the best therapeutic approach because it leads to a model of glucose toxicity. And we think this occurs through several different mechanisms, including disruption of mitochondrial proteins through modification of the enzymes, as well as disruption of the gene expression controlling these pathways, and that this mechanism is consistent in various models of glucose delivery and glycanalylation in mouse hearts, as well as diabetic models in mouse hearts, and that this is consistent with patients. None of this is done in isolation. I've had many great mentors along the road. The MIC-Loop4 model was initially developed when I was a postdoc with Dale Abel. And then the GLICNAC models have been developed since I've been here at UAB, and I've had several collaborators with various human studies, some of which I was able to talk about today, some of which I wasn't. And then most of the work I showed you today was by my postdocs, Jenna, Brahma, and all the bioinformatics that I showed today was done by a former MD PhD student, Mark Pepin, who's now doing his residency at Harvard. And thank you for your attention, and thank you for the opportunity to share our work.
Thank you very much, Dr. Adam. Nice and fantastic presentation as always. So we are going to the Q&A discussion. So for the presenters, if you would like to share your question, so please share your question by directly giving a talk here, by unmute your voice, or you can give in the chat session. So anyway, so before while we are waiting for the question, so I would like to invite Dr. Bambang here as the panelist here. So probably if you have some, if you would like to share your idea, maybe the suggestion or question for both of two prominent speakers here. So please, Dr. Bambang.
Okay, thank you so much, Dr. Jeffy. So Professor Sadoshima and Professor Wende, it was a fantastic lectures. I enjoyed very much. Thank you so much. And I have to disclose myself first. So currently, most of my times was used as a cardiac intensivist in T2. However, diabetic cardiomyopathy and heart failure is part of my long life patient since I did my PhD in Japan 10 years ago. So it really enjoyed to hearing your lectures. So I have some questions and comments for Professor Sadoshima. The first is, I really interesting that you mentioned in some of your previous study that there was some difference about the duration of mitophagy among different kind of exposure. Like if you did the model for pressure overload model by TAC, you present clearly that the mitophagy is all only transient. However, I'm wondering how, if the model is an ischemic model, such as a coronary ligation mice model, so will it be a longer duration of mitophagy, or similar to those happen with pressure overload? So because if I related this to the diabetic cardiomyopathy, we know that the changes in myocardial structure is mostly more similar to those with hypertensive heart disease, but different with the myocardial ischemia. So that's why we call it diabetic cardiomyopathy, because it's independent to the atherosclerosis, right? So that will be my first question. And the second one, from your previous study, you show very nicely that the autophagy in the border zones probably related to the decrease in the infarct size, and then those mechanism leads to the protective effect to prevent the heart failure condition. So I'm wondering whether this might happen in the setting of human with a chronic coronary syndrome with left ventricular dysfunction, that you may notice the recently published a clinical trial, so-called revived BCI-2 trial, that the population is the coronary artery disease patient with left ventricular dysfunction, so with heart failure. However, they convinced that the myocardium is still viable, so they decided to do the PCI to some targeted lesion, coronary lesion. However, as compared to the control with non-intervention, there are no different outcome in terms of mortality and also the effect of recovered EF. You know that we really wish that our patient with heart failure with reduced EF can have some episodes to recover, because the recovery of ejection fraction has been reported to increase the survival and the quality of life of the patient. Probably that's for the beginning of discussion, Dr. Jeffy.
Okay, thank you for your question. And in terms of, you asked whether mitophagy occurs differently in the diabetic cardiomyopathy and pressure overload and myocardial ischemia. And in our experience, duration of mitophagy all different in three conditions you mentioned. And in case of pressure overload, we found that it's very transient. But in case of diabetic cardiomyopathy, it's a little bit prolonged. Even after 20 weeks, we observed mitophagy. In case of ischemia reperfusion, our experience is their activation is very, very transient. And I don't know whether chronic MI, how long it can activate, but I speculate that there are many upstream signaling mechanism which affects the mechanism of autophagy and mitophagy. And we showed in diabetes model, we found that the MST1 is actually activated and try to inhibit. But I don't know whether that is true in other cases. So it also depends upon upstream signaling mechanism. But the idea to reactivate the mitophagy to rescue cardiomyopathy is all same. So we really have to understand what kind of signaling molecules are activated or inactivated in each case. And I think it's a very good question, but we really have to study more, And in terms of second issue, I think, myocardial infarction and after, development of cardiac remodeling, cardiac dysfunction is a major issue in the field. And I really want to find out autophagy activation in humans. And in case of the mice, I believe that the activation of autophagy in border zone is really important. And that not only improve cellular quality control mechanism, of course, you know, in injured myocardium, many protein are degraded or organelle dysfunction are developed. So it's really key to get rid of those toxic materials. And also, most likely, inflammation, infarct resolution is also affected by autophagy, but you know, it's not my expertise. So I really want to find out the connection between autophagy and inflammation. And actually, some people are interested in not only myocyte but also macrophage. You know, the autophagy activation in macrophage is probably important, but more studies needed. And how to maintain the level, I think, you know, the revascularization is probably helpful, but I don't know how that affects autophagy and mitophagy. It's a good subject, and if I have a model, I really want to study, but you know, at this point I don't have any data to you know, discuss this possibility.
Yeah. Thank you so much. How about in fibroblasts? Do you think the mitophagy in fibroblasts will also play important roles?
Yeah, I think in terms of infarct resolution things, you know, we really have to understand both macrophage and fibroblast. And but so far we have you know, the macrophage specific lines, but we haven't done it yet. It's a good point.
Well, I'm waiting for your result then.
Yeah.
Okay, thank you, Dr. Jeffy. Can I ask something to Dr. Wende?
Yeah, of course, please.
Yeah, so thank you so much, Dr. Wende. Very nice lecture. So first, I would like to thank you. I really like your paper, I think a couple of years ago, about you wrote a paper about the guideline for diabetic animal models. So thank you so much, because in my PhD course, I did so many kind of animal models, and finally came to streptozotocin induced diabetes to do my diabetic cardiomyopathy research. So thank you for putting that paper on. From your lecture, I'm interested that you found there was some genes who actually showing dysregulation in the mice models and also in the human with diabetes and also heart failure. So that really a good point to start, I think, to continue your exploration. About the GLUT4, the result of your study showing that the GLUT4 activation then leads to the glucose restoration in the myocardial seems to be have a negative effect for general. So is there any chance to inhibit the GLUT4 activation? That's my first question. And the second one is, from all the multi-nature pathway of diabetic cardiomyopathy, and you focusing on the glucose and also lipotoxicity, I'm wondering, do you have any chance to see the myocardial lipotoxicity in a human with diabetes, for example, by using MRI spectroscopy or something? Probably you can share your experience. Thank you.
No, thank you for those questions. So for the first one, we have looked at glucose knockouts before. And this was mostly work I did still as a postdoc with Dale, where we knocked out either GLUT1 or GLUT4. And unfortunately, when you knock those out, it also leads to dysfunction. So overexpressing leads to dysfunction, knocking out leads to dysfunction. In the case with the GLUT4 knockout, what we found was that loss of GLUT4 in either pressure overload-induced hypertrophy or exercise-induced hypertrophy led to a crosstalk signaling to fatty acid utilization, and it further enhanced that lipotoxicity, where we actually saw enhanced ceramide signaling. So taking away the heart's ability to use GLUT4 did that. Now, we did not look at it directly in diabetic cardiomyopathy. We only looked at it in pressure overload and exercise. So we haven't exactly answered your question. And then to your second point, unfortunately, I'm a PhD, so most of the human stuff I have to get through collaborators or through the literature. Even in the human studies that we have done, almost all of that is de-identified tissue that's been in the freezer, so I don't have access to much patient data on them. So unfortunately, I don't have the answer to that.
I see. So following your response, if you have some tissue sample, the myocardium from failing heart with diabetes, so what will be interesting findings in terms of lipotoxicity?
Yeah, so I think almost all of what we've done is transcriptomics. We've done a little bit of histology on them. We have not done any metabolomics on them, mainly just by the way the tissue is processed. It's not very consistent. So I would not want to waste my money doing metabolomics on it. But from the transcriptional profiling, I will say that a lot of the gene expression changes consistent with the Venn diagrams I showed were consistent with disruption of fatty acid utilization. So I think the gene expression would at least be consistent with those. The question is what's the downstream function? What are the proteins? It would be great if we could do all the different omics. I am currently doing one project with Heiko Buger in Graz, Austria, where we're trying to get to that answer. He's a clinician and has access to quite a few and fresher hearts. So we're trying to get to some of that metabolomics question. So TBD. And I will say of potential interest to you with that collaboration is we're focused specifically on diabetic patients with preserved ejection fraction versus diabetic patients with reduced ejection fraction. And we're hoping to get that paper submitted ideally by the end of the year, but it's been, we're trying to add in some single cell sequencing as well. So when it comes out, it should be a pretty big story.
Oh, great. I look forward for it. Thank you so much.
And I actually had a follow-up question for Dr. Sadoshima, if that's okay.
Yeah, of course.
Yeah, you were I really enjoyed your talk as always.
Thank you.
The one thing I keep on getting pushed on now with technology is single cell, and you were kind of getting to that at the end as far as the fibroblasts and macrophages. Do you think some of that intermittent initial induction versus a sustained induction in your whole tissue analysis reflects like the M1 to M2 initial phase versus cardiomyocyte changes? Does that question make sense?
Yeah. So everyone asks, the cell type specific response, and we are trying to do. We have a program in single cell sequencing analysis because myocyte is too big, and so we try just started the single nuclear sequencing, and also there's a way to differently barcode single cells and different technology, but we so far we haven't got any result yet. So I think I briefly mentioned the previous question. I'm very much interested in autophagy in macrophage and so that it may give us some clue how information process is affected. But good question, but so far I just can't answer to your question yet.
Okay,
Thank
no,
you.
that's fine. And the other follow-up, and this is, so with the DNOGA GLICNAC mice we have, we're currently collaborating with Martin Yun here at UAB, and we do have some evidence of the autophagy machinery being O-GLICNAC-related, so you may want to take a peek there as well.
That's great. So if we can collaborate anytime, I'm ready. Yes.
Sounds good.
Thank you very much. So we still have a few time for the discussion session. So we are inviting for the participant. If you have any question here, please share your idea. So we have so thank you very much. So I saw Dr. Renan as the head of cardiovascular department and also other cardiology staff. Thank you very much for joining, and also the Dr. Nani as the head of sport medicine. So yeah, please share your question here. So anyway, if you don't mind, so I would like to ask also to Professor Sadoshima and also Dr. Adam. So maybe I would like to start with Dr. Adam. So I was interested with the cotton body. So as you know, maybe the paper was published in the JACC 2021, maybe from Groningen University, talking about the therapeutic potential of cotton body in cardiovascular patient. So I was wondering that at that paper, they mentioned that the cotton body administration has a beneficial beneficial strategy for the cardiovascular disease. So what do you think? Is that the ketogenic diet, or maybe some diet intervention like a fasting that can induce the fatty acid oxidation as a beneficial function for the diabetic cardiomyopathy patients? And also the second question is similar to the Professor Sadoshima. So as we know that the fasting, if you're talking about the fasting, can induce the autophagy. So what do you think about the role of the fasting, or maybe the cotton body in the autophagy, metaphagy in diabetic cardiomyopathy? So please, we can start from the Dr. Adam first.
Okay. No, I think that's a great question. I think the literature has a lot of controversy out there. I was involved with a point-counterpoint on this very question, because I think the literature supports both ways. And I think it has to do with timing. So I think there's definitely benefits in intermittent fasting and ketogenic diet for weight loss. Where it gets a little more complicated is when you overlay it on a diabetic patient. So not work I've done, but we have a nutrition and obesity research center here, or our NORC, and we had our symposium last week. And they were talking about the different intermittent fasting diets. And one of the things that really struck me is that although this whole, I don't know if you've heard of the 8-hour diet, where you pretty much eat all your meals between an 8-hour window and then fast the rest of the time, which leads to a slightly ketogenic state and weight loss. What they showed in their literature review is that it was not beneficial in the diabetic patients. So although it may be beneficial in obese patients, for some weird reason, it does not appear to have the same benefits in the diabetic patients as it does. Now, again, this isn't my work. I don't know for sure. But I do think we have to be careful as far as pushing these systems, as far as comorbidities and whatnot.
Thank you very much. Yes, thank you first. So for the second question, maybe, thank you, Professor Sadoshima.
Yeah. So just Adam mentioned that the intermittent fasting is not so beneficial in humans, but in terms of animals. It is shown that autophagy genes are regulated in a circadian manner, and so there's a recent Nature paper that if the intermittent fasting is designed to enhance autophagy mechanism during the night, it may be beneficial. And I like that paper because it's known that the cardiovascular incidence is higher in the morning because of sympathetic nerve drive. So if you have fasting during the night when autophagy gene is really high, we can enhance the autophagy mechanism in the morning. So that can precondition the heart. So I think, maybe, if you focus on some cardiovascular incidents, and then if you impose fasting during the night, I suspect it should be good, but it's a really human study, so you never know. That there are many complicated factors. So that's only my opinion.
No, I think you're right, and consistent with that NORC symposium, what they're finding, this is in-house where they're controlling the calories, and they found very specifically that if you did your 8-hour window of eating in the morning, you had more benefits in weight loss and insulin sensitivity than if you did your 8 hours in the evening. So I think you're definitely right that getting it in the morning and having the prolonged fast at night helps, probably through autophagy, I would guess.
Yeah, thank you very much for the answer. So the time is running out, so maybe any last words, Dr. Bambang, so to be mentioned here.
No, I think I would like to thank you again to our both speakers who already widen up our horizon on this topic. And so, yeah, hopefully starting from this point, we may continue to the potential collaboration with our research center. So, Dr. Jeffy, I saw our head of department, Dr. Renan. Probably Dr. Renan would like to have a comment.
Yeah, thank you very much. I very much enjoying this meeting. Hopefully, as Dr. Mama mentioned, we can have more collaboration on this. We do have lots, especially in our center, National Heart Center. We have lots of patients with various diabetic and cardiomyopathy stages, as well as other comorbidities. Hopefully, we can have more forum in this, and also again to have starting like research collaboration on these related topics. Can I have a question, Dr. Bambang and Dr. Jeffy?
Yeah, please.
Yeah, probably this is about because some of our audience is a clinician and related to diabetes and also obesity. Just to know what's current status and understanding on the benefits of bariatric surgery. There are some evidence showing that they do have benefit in terms of heart outcome, such as reducing future cardiovascular events and even lower mortality. But the better than those who still on obesity, but the beneficial is not will not be similar with those without obesity from beginning. So can our faculties here have a comment on that, please? Thank you.
Yeah, please. Maybe Dr. Adam or Professor Sadoshima. Any suggestion?
I mean, I followed the literature. I think it's very interesting as far as some of the signaling that can go on, but unfortunately, it's outside my area of expertise. So,
yeah, I think the evaluating autophagy in humans is a big issue, and we are trying to find out biomarkers, but even animals. Autophagy analysis is a little bit complicated. So yeah, I think if we have some animal model, we can speculate what's going on. But surgery is actually effective, right, in terms of inhibiting. So it's probably important to find out how it affects metabolism and signaling mechanism, so that we will know more about the mechanism. But I'm sorry, I don't know much about it either.
Okay, thank you very much, Dr. Adam, also Professor Sadoshima. Okay, I'm sorry to finish this webinar because it's almost 9 p.m. in Indonesia. So thank you very much, Professor Sadoshima. Thank you very much, Dr. Adam. Very nice presentation. And also thank you very much, Dr. Bambang. Thank you very much, Dr. Rena, Professor Badrul Hagar, and also other participants. I hope we can meet again in the very nice presentation or maybe other webinar in the future. So I would like to finish this webinar. Thank you very much and have a very nice day. Thank you very much. Assalamualaikum.